These findings of inflammatory compounds in animal foods and fermented foods therefore suggest that apparently unspoiled foodstuffs may nevertheless contain at some point in their preparation and processing a sufficient microbial load to release endotoxin receptor stimulants into their growth environment. This notion is supported by many previous studies showing that certain commonly consumed foodstuffs can contain a high bacterial load before cooking, such as fresh hamburger, which has often been shown to contain approximately 100 million bacteria per quarter pounder. Notably, however, the purpose of the present study was not to examine the microbial quality of each foodstuff, since the pathogen-associated molecular pattern, biological activity, is retained independently of bacterial viability or cooking, meaning the bacteria can be dead, the bacteria can be cooked, but their endotoxins are still there. You can boil meat for 2 hours straight, dip it in an acid bath, like our stomach, and expose it to digestive enzymes. Bacterial endotoxins were found to survive both cooking and our body's best attempts at acid and enzyme digestion. This led them to speculate that the occasional ingestion of meals high in bacterial endotoxins could promote transient mild systemic inflammatory episodes that predispose subjects to the development of atherosclerosis and insulin resistance, which can lead to diabetes. The animal fat may play a profound role in the pathogenesis of this after-meal inflammation by increasing the absorption of the endotoxins. That's where the animal fat comes in, since endotoxin has a strong affinity for the saturated fat transport system through the gut wall and into the bloodstream. So animal fat may play a role in boosting endotoxin absorption, but the primary reason all those studies show increased inflammation from animal foods, but not from most plant foods, may be the load of dead bacteria in animal products, which release endotoxins that are absorbed into our system, leading to the endotoxic inflammation we see after meat, egg, and dairy consumption as well. So, now that we know what's going on, what do we do? From a 2012 follow-up, well, the most obvious solution to this metabolic endotoxemia appears to be to reduce saturated fat intake, which in the US is mostly from cheese and chicken. But the researchers fear the Western diet is not conducive to this mode of action. It's difficult for patients to comply with this request. Probiotics have moved from the field of alternative medicine into the mainstream slowly but surely over the past decade. The best evidence we have is for the prevention of antibiotic-associated diarrhea and the treatment of gastroenteritis. Antibiotics administration is followed in up to 40% of cases by the appearance of diarrhea. But, for example, you may be able to cut the risk in kids in half by administering probiotics along with the antibiotics. Which kinds and how much? Lactobacillus rhamnosus and Saccharomyces boulardii appear to be the most effective strains, and studies using more than 5 billion live organisms appeared to achieve better results than those using smaller doses. The importance of correct dosing cannot evidently be overemphasized. For example, in adults, going with 100 billion organisms seem to work nearly twice as well as 50 billion in preventing antibiotic-associated diarrhea. The second well-established usage of probiotics is in the treatment of acute infectious diarrhea, shortening the duration of symptoms by about a day. But we still don't know what the best probiotic doses and strains are. Studies have used between 20 million organisms a day to 3 trillion, and there are thousands of different strains to choose from. And even if you wanted a particular strain, odds are the label is lying to you anyway. Less than a third of commercial probiotic products tested actually contained what the label claimed. About half had fewer viable organisms than stated, and half contained contaminant organisms, including potentially pathogenic ones, as well as mold. Now, ideally, we'd repopulate our gut with the whole range of natural strains, and for serious infections, this has been attempted starting back in 1958. Why not give people a fecal enema take the full complement of gut bacteria from a healthy colon and stuff it into an unhealthy colon. Or you can go the other route and administer the donor stool through the nose. Evidently, this route of administration saves time, is cheaper, less inconvenient for the patient. Preferred stool donors, in order of preference, were spouses or significant others, family members, and then anyone else they could find, such as a medical staff member. What you do is first pick a nice, soft specimen, whip it up in a household blender until smooth, a little Vitamix action, put it through a coffee filter, and then just squirt the stool up their nose through a tube and into their stomach. Don't try this at home. How receptive were the patients to this rather unusual smoothie recipe? None of the patients in this series raised objections to the proposed stool transplantation procedure on the basis that it lacked aesthetic appeal. However, since production of fresh material on demand, not always practical, researchers up in Minnesota recently introduced frozen donor material as another treatment option, all described in great detail in the latest review on the subject out of Yale, entitled The Power of Poop.
We know that many plant foods benefit our health. Scientists now believe one reason for this lies with the gut microbiome, the bacteria in your intestines. Your microbiome is nourished by meals like this, rich in one type of dietary fiber called resistant starch. Resistant starch can't be digested by your body, but instead becomes food for your gut bacteria. Most starch is easily digested. Starch is dissolved in the small intestine and then absorbed by your body, providing you with energy and nutrients. The remaining non-digestible portion is called resistant starch. The resistant starch continues its journey through your gut and arrives at the large intestine. We see that the resistant starch has become exposed to the healthy bacteria of the gut microbiome. This species of bacteria specialize in breaking down the resistant starch. This breakdown process provides the bacteria with the fuel they need to survive. As they use the starch for energy, they release small carbohydrate molecules. The neighbouring bacteria feed on these carbohydrates. As the bacteria feed, they excrete even smaller molecules as waste. One of the final waste products is called butyrate, your body and energy. As the butyrate builds up, it is absorbed by the large intestine. The presence of butyrate encourages blood to flow into the vessels of the large intestine, keeping the tissue healthy. If your diet includes enough resistant starch, these cells will use butyrate as their main source of energy. Here, we can see the molecular surface of one of the intestinal cells. The surface is covered in special proteins that actively pump butyrate molecules into the cell. Once inside, they can be harvested for energy. In addition, butyrate has other benefits. Intestinal cells are sensitive to DNA damage caused by environmental factors. This cell's DNA has been damaged, resulting in a mutation. More damage could accumulate over time as the cell divides, which could lead to colorectal cancer. But A steady supply of butyrate allows the DNA damage to be more easily detected, and the cell can activate a suicide program in response. Because the damaged cell destroys itself, it can't progress to form a cancer. A starved microbiome is unable to protect you from cancer. By eating foods rich in resistant starch, you can nourish your microbiome and improve your health.
When researchers at the Emerging Pathogens Institute recently ranked foodborne pathogens to figure out which was the worst, number one on their list was Salmonella, ranked the food poisoning bacteria with the greatest public health burden in our country, the leading cause of food poisoning-related hospitalization, and the number one cause of food poisoning-related death. Where do you get it from? Well, I've talked about the threat of eggs. According to the FDA, 142,000 Americans are sickened every year by egg contaminated with Salmonella. That's an egg-borne epidemic every year. But Salmonella in eggs was only ranked the number 10 worst pathogen-food combination. Salmonella in poultry ranks even worse, the number 4 worst infected food in the United States, in terms of both cost and quality-adjusted years of life lost. In terms of the burden of human salmonella poisoning attributable to various U.S. foods, eating chicken may be 8 times riskier than eating eggs. Due to strengthening of food safety regulations under the Clinton administration, the number of Americans food poisoned by chicken every year dropped, from about 390,000 a year to 200,000, and rightly hailed as a significant accomplishment. So now eating chicken only sickens about 200,000 people in the U.S. every year. But isn't that a bit like some toy company boasting that they've reduced the amount of lead in their toys, and so are now poisoning 40% fewer kids? Not exactly something to boast about. And the numbers have since rebounded back upwards. In the late 90s, human salmonella cases have increased by 44% since then. The rebound in incidence of salmonella infection in the United States is likely a result of several factors, but one important risk factor singled out is eating chicken, as the proportion of chicken carrying salmonella infection has increased. When people think manure and meat, they typically think ground beef, but when you look at E. coli levels in meat, which is considered an indicator of fecal contamination, sure, there's fecal matter in about 2/3 of American beef, but that number is greater than 80% fecal contamination in poultry, chicken and turkey. Why have we seen a decrease in the Jack-in-the-Box E. coli 0157, but not chicken-borne salmonella? In the last decade or so, the infection of beef and subsequent children 30%. But not only has salmonella not declined in the past 15 years, it's actually increased lately. One reason is that there was a prohibition of contamination with the deadly E. coli in beef. What a concept! So selling contaminated beef is illegal. Why is beef laced with E. coli contaminated fecal matter considered adulterated, but chicken laced with salmonella contaminated fecal matter A-OK? It certainly kills more people than the banned E. coli. It all goes back to a famous case in 1974, when the American Public Health Association sued the USDA, saying, wait a second, you can't put a stamp of approval for wholesomeness on meat contaminated with salmonella. What could the USDA possibly say in meat's defense? As relayed by the circuit judge, the USDA pointed out that there have been salmonella outbreaks linked to dairy and eggs, for example, too. So, since there are numerous sources of contamination which might contribute to the overall problem, it would be unjustified to single out the meat industry and ask that the department require it to identify its raw products as being hazardous to health. That's like the tuna industry arguing, there's no need to label cans of tuna about mercury levels, because you could also get exposed to eating thermometers. The D.C. Circuit Court of Appeals upheld the meat industry position, though, arguing that you can allow potentially deadly salmonella in meat because, and I quote, "American housewives are normally not ignorant or stupid, and their methods of preparing and cooking of food do not ordinarily result in salmonella food poisoning." What? That's like saying, minivans don't need seatbelts in the back seat because soccer moms don't ordinarily crash into things. Now, 39 years later, 200,000 Americans sickened every year by salmonella that continues to be legally allowed in chicken.